Hello. Thank you to the organizers for inviting me to give this talk, and thanks to all of you for tuning in. My name is Jamie Neer, and I'm going to be talking to you today about how to use the FID-A toolkit for MR spectroscopy data processing and simulation. So first, a brief outline of the talk. I'm going to start by giving an introduction to FID-A and what it does and what it doesn't do. And then I'm just going to walk through some practical examples of how to use FID-A. So I'll talk about how to download and install the software, how to process MRS data, how to do data quality assurance in FID-A, and then a little bit about how to use the RF pulse analysis tools. And lastly, I'll talk about how to simulate MR spectroscopy experiments in FID-A. So first, what is FID-A? Well, FID-A is an open source Matlab-based software toolkit for MR spectroscopy simulation, data processing, as well as RF pulse design and analysis. FID-A gives us a framework to access, manage, and visualize all aspects of your MRS data. The source code is fully open and it's accessible on GitHub at the website URL shown here. A few words about the features of FID-A. So it's written in MATLAB and mostly command line based. Most common spectroscopy processing operations can be done with a one-line command. And this gives you maximum control of your data with minimal user input. The FID-A toolkit is made up of 5 different toolboxes. The input-output toolbox for reading and writing MRS data to and from different file types. The processing toolbox for data processing. The simulation toolbox for simulation of MRS experiments. The RF pulse toolbox for RF pulse design and analysis. And the example run scripts, which are collections of functions from other toolboxes but organized into useful pipelines. Every function name in FID-A starts with a suffix followed by an underscore, followed by the actual function name, and the suffix indicates which toolbox each function belongs to. So functions in the input-output toolbox start with an IO suffix, and functions in the processing toolbox start with OP. The simulation toolbox has the SIM suffix. RF pulse tools start with RF, and example run scripts start with run. A little more about the features. So MR spectroscopy data typically has multiple dimensions. So when you get the raw data off the scanner, it'll have multiple RF coil channels from all their different receivers. It usually has multiple averages, and it might even have multiple subspectra. For example, in the case of spectral editing data, you might have an edit on and an edit off subspectrum. So FID-A is designed specifically to handle this type of multidimensional data and to manage these dimensions and be able to manipulate them. It has a large library of processing tools and simulation tools, and the data are stored in a structure format that contains not only the data itself in both the time domain and frequency domain, but also contains all relevant header information. There's also a library of run scripts, which has pre-implemented processing pipelines and simulation pipelines, as well as tools for generating basis sets. And lastly, because it's all coded in MATLAB and it's open source, if users who have experience with MATLAB can easily modify the codes to suit their needs. Next, I'm going to show you just a list of all of the different tools or functions that exist in MATLAB in each toolbox. So this is a list of all of the functions in the input-output tools, and I've organized them here by vendor type. So here are tools for reading and writing data in Siemens format, Brooker format, GE, Philips, Varian Agilent, and then you can also read and write data in the formats of analysis softwares like JMRUI, LC Model, and Vespa. Here's a list of all the processing operations in FID-A. These I've kind of categorized by sort of different types. So these are dimensionality manipulation, where you might be combining different dimensions or removing dimensions from your data set. These are frequency and phase shifting operations, filtering operations, math operations like adding and subtracting spectra together, truncation, so that would be kind of chopping your spectrum into different bits along the frequency domain, for example, basic peak fitting operations, quality assurance operations, spectral registration for aligning spectra, plotting tools, and I guess other tools that are also considered processing operations. We also have a simulation toolbox. So in the simulation toolbox, you have basic spin operators. So these are basically the simple operations that you can apply to your spin system. And then on the right are pulse sequences, both ideal and shaped pulse sequences that are essentially collections of the basic spin operators to make the common pulse sequences that we use in vivo spectroscopy. Here are the tools in the RF pulse toolkit. So we have block simulation tools for simulating the shapes, the frequency profiles of RF pulses. And on the right, RF pulse design tools, so for designing different types of RF pulses. And lastly, here are the example run scripts. So we have these are functions that have been strung together into a pipeline for processing here on the left, simulation pipelines, and quality assurance pipelines, so measuring quality of your data. What are some things that FID-A does not do? Well, first of all, it doesn't have a graphical user interface. It's mostly designed as a command line tool. It doesn't support spectroscopic imaging at the moment, although this is something that's development is in progress. It also does not currently support X-nuclei spectroscopy or X-nuclear spectroscopy. This development is also in progress. And finally, it's not really a standalone piece of software because it requires a Matlab license, and it also requires certain Matlab toolboxes to be installed, such as the machine learning, statistics and machine learning toolbox, or the statistics toolbox in older Matlab versions, as well as the parallel computing toolbox. So now that I've given a brief introduction of FID-A, I'm going to go through some practical examples of how to do certain things in FID-A. And we're going to start by showing how to download and install the software. So to download, there's two ways. You can do the basic installation, which involves going to the GitHub website. And once you're there, you're going to click on the clone or download button. And from there, you're going to click the download zip button. And this is going to download a zip file that contains the entire toolbox. Once you've got this, you're going to unzip it somewhere on your computer and move the resulting folder into the desired location. Another way to download and install FID-A is to use a Linux terminal. So if you have a Linux computer or a Mac, you would open your Linux terminal, and then from there you're going to first navigate into the directory where you want your FID-A copy to be located, and then you're just going to type git clone and then type the URL where on GitHub where FID-A is located. And once you type that, it's going to download the whole package for you. A quick note is that one of the things included in FID-A is a directory of example data. And unless you have the git LFS package installed, which is the git large file storage package, it won't properly download these example data. So try and install git LFS before you start, and that way you'll get all of the example data when you run this clone method. So now that you've downloaded the software, you just have to open Matlab, and you're going to add the FID-A directory and all of its subdirectories to your path using the following function or call. So you type add path, gen path, and then the directory where you put the FID-A folder. In my case, it was on my desktop. So you've done that, and now you're done. You're ready to use FID-A and all of the functions inside. So we've done the installation. Next, let's talk about how to load an MRS dataset and then manually process it in a step-by-step fashion. So on this example, we're going to use Siemens Megapress data. Since we're at the editing school, I thought we would do an example with some spectral editing data. This data is actually found within the FID-A download directory. So this is the FID-A directory that you downloaded in a subfolder called example data /Siemens /sample01 megapress. This is what will be downloaded with the package, assuming you have the git lfs installed. And here we have a folder with water suppressed megapress dataset, as well as a water unsuppressed megapress dataset, and these are in the Siemens Twix format. So I keep the water suppressed data and the water unsuppressed data in separate folders for reasons that I think you'll see later, but this is sort of the convention that I use in FID-A, is just to keep them separate, and with the file names, or the folder names rather, being identical except for an _W corresponding to the folder where the water unsuppressed data is located. So if I want to load this data into FID-A, first I'm going to navigate to that directory where the example data is kept. I'm going to load the water suppressed data using this function io load spec twix, followed by the name of the .dat file, or the Twix file that I want to load. And then I can also load the water unsuppressed data using the exact same command, but pointing now to the water unsuppressed data. And once I've done that, I'm going to have two structures in my workspace, the water suppressed data and the water unsuppressed data. And these are the names that I gave in my function call. Those are the output names that I gave. Next, maybe we want to look at the data structure. So to do this, we're going to type the variable name at the command line and just hit enter. So in this case, the variable is called raw, the water suppressed data. And I'm going to look at the structure. So the structure has a lot of different fields. The main ones here are FIDs and specs. This contains the time domain and the frequency domain data respectively. And you can see that these arrays have multiple dimensions, and we're going to see what those dimensions are. So it's an array that has 2080 by 32 by 80 by 2 elements. And to find out what those are, I'm going to query this field here, which is called dims. So I'm going to type raw.dims and hit enter. And when I do that, it's going to tell me which dimension number corresponds to the different dimensions of this structure. So the time dimension is the first dimension. That means that this 2080 points are along the time dimension, or in the case of the frequency domain, that would be the frequency dimension. And the second dimension is the coils dimension. So we have 32 elements in the coils dimension. The third dimension is the averages. So that means we have collected 80 averages of data. And the fourth dimension is the subspecs dimension. So this would be the edit on and edit off scans in that dimension there. And so this is how the data is stored. And the rest of these parameters in the structure are mostly to do with header information, such as the date that the scan was acquired, transmitter frequency, the dwell time, and so on. The next step in processing this sort of in a step-by-step way might be to combine the RF channels. So I have 32 channels, and I don't really want all 32 channels to be separate. I want them to be all combined. So to do that, I'm going to use a function called op_combine receivers, and I'm going to pass in both the water suppressed data and the water unsuppressed data. And I'm going to have two outputs, which are going to be the water suppressed and water unsuppressed data, but with the coils, the RF channels combined. So after I've done that, let's query the output out1. And if I look at it, now its dimensionality has been reduced. Now it only has 2080 by 80 by 2. And if I look at the value of this dims parameter again, this dimension parameter, it's showing me that the time dimension is the first dimension. The coils dimension has now gone to 0, meaning that the coils, there are no multiple coil elements left. And the averages dimension is now the second dimension, and the subspecs dimension is the third dimension. So you can see this combine receivers command has combined those 32 receiver channels and gotten rid of that dimension of the array. Okay. So let's say now we want to plot the spectra. So if I want to plot the spectra, I'm going to use a function called op_plot FID. This will plot the data in the time domain, and I'm going to plot out 1. When I do that, it's going to ask me, which dimension would you like to display? Because we have 80 averages and 2 subspectra, and it can't plot all of that at the same time. So it says, do you want to display the averages or do you want to display the subspecs? So in this example, let's say I choose to view the averages. So I'm going to enter the number 2, and it's going to plot me all of the averages in the time domain for the first subspectrum. And there you have it. We can also plot the data in the frequency domain, and to do this, we're going to use the function op_plot_spec. Again, I'm going to plot the same data set. This time, again, it asks, do you want to plot all of the averages or all of the subspectra? Let's see what it looks like when we ask to plot the subspectra. So that's number 3 in this case. And here it's showing the first average, but both subspectra, so the edit off scan and the edit on scan. So there's our plotting functions, op_plot_fid and op_plot_spec, for plotting the time domain and frequency domain data. Now let's do some more processing to the data to get the final difference spectrum at the end. So the first thing we might want to do is do some spectral registration to get rid of frequency and phase drifts across the averages. So we're going to use this function called op_align averages and apply that to the last coil combined data that we had. And each time I do this, I'm making a new output. So the first output was called out1, the next one I'm going to call out2, and as I go along, I'll rename each time out3, out4. So let's say I do that, and now after I've aligned the averages, I can do op_plot_spec, and I can see all of the averages aligned together. Now you can see that any drift that was there seems to have been corrected. Okay, and the next step I want to do is to combine those averages. So I'm going to average them all together using the function op_averaging. And when I do that, now my data set has been reduced from 80 averages just down to 1 single scan. But I still have 2 subspectra. So before I subtract the subspectra from each other, I might want to align them. And so I want to align them using this function op_align MP subspecs. That stands for megapress subspecs. And so I'm taking the output of the previous command, and passing it into op_align MP subspecs to get a spectrum where the on and off scans have been aligned and are ready for subtraction. Now I'm ready to subtract them. So I'm going to subtract the megapress subspectra using op_combine subspecs. So I'm going to pass in out4, and I'm specifying that I want to take the difference between them. And once I've done that, then I have a single spectrum, which is my final difference spectrum. We're calling it out5. Maybe before I view it, I might want to do a little bit of zero order phasing and filtering of the data. So I want to automatically phase it using this function op_autophase. Here I'm showing how to do both zero order phasing and filtering in a single line. So I'm calling the output out6, and I'm going to first phase the output using this op_autophase function. And what I do here is I enter the previous output, which was the output that had the subspectra combined. I'm going to phase on a peak that's between 1.8 and 2.2 ppm, which is the NAA peak. And I'm going to phase it such that it's a 180 degree phase. So that means it's going to be inverted. And if you remember from some of the earlier talks, you'll know that in a megapress difference spectrum, usually we have the NAA peak inverted. That's why I give this phase a 180 degree phase. And then I'm taking the output of that, I'm passing it into this op_filter function, which is essentially going to apidize the spectrum by a 2 Hertz exponential filter. So this is phasing and filtering all in one step. And then at the end, I'm going to plot the result using OP plot spec. And here's my result. So I have my NAA peak, which is inverted, as we would expect, and I have my GABA peak at around 3 ppm, which is here. Great. Maybe at the end, now that I have a fully processed spectrum, I want to write that spectrum into an LC model format so that I can analyze the data in LC model. So I'm going to use this function io_write LCM, which is going to take that output. Actually, that should say out6, or it could be out5. It doesn't really matter. You're going to take one of these two outputs. You're going to turn it into a text file that can be read by LC model using io_write LCM. And the last term here is the echo time that you're going to put in milliseconds. So that's how to process a data set manually, and you can see it was a very step-by-step process. This is sometimes useful if you really want to tailor your processing on a case-by-case basis. But for the most part, if you're running a large study, you want to apply the exact same processing pipeline to many data sets, and that's where it becomes handy to have an automated processing pipeline. So this is a script where all of those steps I just showed you are embedded into a single script and can be applied to any data set, and can be applied to many data sets in a row to get the exact same processing on each data set. So these are called the automated processing pipelines, and it's very fast and efficient to run your processing this way. So these are found in the example run scripts directory, and I'm going to show you a script called run megapress proc _auto, which is a script for processing the megapress data. And it's similar to the steps I just showed you, but it's all done in a single step. So the way you run it is you're going to call this function called run megapress proc _auto. You're going to apply it to the directory the name of the directory that contains your water suppressed data. And as outputs, you're going to put you're going to have the difference spectrum, the sum spectrum, the edit off and the edit on spectra, as well as the water unsuppressed spectra. So if I run that, it spits out a lot of kind of text, and some of that is useful, some of that's not so useful. And at the end of it, it's going to give you these outputs that you asked for: diff, off, on, and sum, and water. And these are all of the outputs from the processing pipeline. And so I can view the result using OP plot spec as I did before. So let's say I want to view the difference. Let's say I want to view all of them. So previously when I showed you how to use OP plot spec, I just put in a single structure as the input. But if I pass in a cell array, which is defined by these curly brackets, I can plot multiple objects at the same time, or multiple structures at the same time. Here I'm going to plot the difference, the sum, the edit off, and the edit on spectrum all at the same time using this OP plot spec by passing them as a cell array. And I'm going to give it a legend so I can see what each trace represents in terms of its color. So when I type that into Matlab, I'm going to get a plot that looks like this, and you can see clearly the blue is the processed difference spectrum. We also have the sum spectrum, the edit off, and the edit on spectrum. And so all of these were produced by the single processing script run megapress proc auto, and then they're plotted using OP plot spec. So you can see this is a really handy one-line command to process an entire megapress data set. Another thing that gets produced by this processing script is a report file. So if you look in here, if I open up the megapress directory, it's going to create a report directory, and inside there's a report.html file that we can look at. So this report file shows us the results of the different processing steps, such as the coil combination step, the step for removing motion corrupted averages, the step for spectral registration, and then the final results, both the edit on and edit off processed spectra and the difference spectrum. Next, we'll talk about data quality assurance in FID-A. So after you've processed your MR spectroscopy data, it's a good idea to check the spectral quality. And the two most important and unbiased spectral quality metrics are the line width of the data, or the line width of the spectral peaks, and the signal to noise ratio. So we're going to calculate the line width using a function called op-get-lw. This function calculates the line width of a peak in the spectrum. By default, it's going to search around 4.65 for the water peak. So if we just pass in the water unsuppressed data, it will automatically fit the water peak to a Lorentzian line shape and then estimate the width of that line shape. In this case, it's telling us that the line width is about 5.8 hertz. Another thing we can do is we can select a different peak to measure. Here, if we want to measure the NAA peak, we can pass in the difference spectrum and tell it to look between 1.9 and 2.1 ppm. That's where it's going to find the NAA peak, and it'll fit that peak, which here now is an inverted NAA peak, again to a Lorentzian function and estimate the line width. And again, it's telling us that our line width is now 3.3 hertz for the NAA peak. Lastly, we might want to calculate the signal to noise ratio. So we're going to use a function called op_get-SNR. This function searches for the NAA peak by default between around 1.9 and 2.1 ppm, but you can change the range to search for any peak you want. But in this case, we're passing in the difference spectrum, and it's telling us that the signal to noise ratio for this difference spectrum is about 278. So that's data quality assurance. Next, let's talk about how to load and analyze an RF pulse waveform. So if you navigate to the RF pulse tools directory, you can find a directory called RF pulses, which contains a few sample RF pulses in various different formats. We can load one of these using the function io_load_RF_waveform, and we're going to load this time the sample refocusing pulse.pta, which is it's a refocusing pulse waveform in the Siemens PTA format. And we're going to pass in this ref argument to tell the function that we're expecting a refocusing pulse, and we're calling the output refoc. So there's the command. I'm going to now view the structure by typing refoc, and what you can see is that it has a few different fields, but the first field is what contains the waveform, and these other fields are just sort of header information about the RF pulse. I can plot the amplitude and phase waveforms of the RF pulse using the function rf_plot waveform. And when I do that, I can see that this particular pulse has an amplitude waveform that looks something like a sync pulse, and it has a phase waveform that alternates between 180 degrees and 0 degrees at each of the different lobes of the sync. I can also simulate the frequency profile of this pulse, or the inversion profile, using a function called rf_blocksim. I'm going to pass in the RF pulse itself, as well as the duration of the pulse in milliseconds. And when I do that, I get a plot of the X, Y, and Z magnetization profiles as a function of frequency. And the most interesting one here is probably the Z magnetization, which starts at 1 outside the band of the pulse, where the magnetization is pretty much unperturbed, and then inside the band of the RF pulse you get a full inversion all the way to mz equals -1. So that shows us quite nicely the inversion profile of this particular pulse. And lastly, I just want to walk through a few examples of how to generate simulated MRS data using FID-A. The goal here is to estimate the spectral shape of a particular metabolite or spin system under a particular pulse sequence or acquisition. And this is important because we need this type of information often for spectral quantification, especially if we're using an LC model type of analysis. So FID-A is equipped with a list of metabolite spin systems that's built in, and each one of these metabolites has a set of chemical shifts and coupling constants to describe it, and that we can use to simulate it. So let's have a look at these. If we go to the simulation tools directory within FID-A and slash metabolites, you can see a list of .mat files, and these contain the spin system definitions for each metabolite that's stored inside the FID-A. And let's look at the lactate spin system, for example. So we can load that using load lac, and if we do that, we'll end up with a variable in the workspace called syslac, which is a structure. And if we type syslac, we can see that it has 4 fields, including J, which is the coupling constants, and shifts, which are the chemical shift values. And I can look more closely at these. So if I type syslac.shifts, I can see that there are 4 protons in the lactate spin system, and their chemical shifts are 4.09 ppm, and then there are 3 identical protons with chemical shifts of 1.31 ppm. I can also look at the coupling constants by typing syslac.j, and here I get a 4x4 matrix that tells us the coupling constant between each proton and every other proton. So in this case, we have a coupling constant of about 6.9 hertz between the first proton and the second, third, and fourth protons. As I mentioned earlier, the FID-A simulation toolbox consists of a list of basic spin operators such as excitation and readouts and rotations, as well as a list of pulse sequences. And so these basic spin operators are what form the building blocks of these pulse sequences, and we can use these to do lots of different types of simulations. The very simplest pulse sequence in the list is called the pulse and acquire sequence, or sim1pulse. And to use this sequence, you need to supply the number of points you want in your spectrum, the spectral width, the magnetic field strength, the line width of the desired spectrum at the end, and the spin system that you want to simulate. And if you look into the code of this function, there is not much to it. There just is the sim Hamiltonian function, which essentially initializes the spin system and Hamiltonians, and then simply the excitation pulse followed by a readout. It really is just the excitation pulse and an acquisition. And so we can actually show how that function looks if we apply it to the lactate spin system. And we call it by saying out equals sim_1pulse, open bracket, and then the input parameters that we want, and we're applying it to the lactate spin system. And the output structure that we get at the end looks a lot like the data structures that I was showing you earlier. And we can plot that using the op plot spec command. And what we can see is a simulated lactate spectrum following a single pulse and acquire acquisition. What other pulse sequences do we have? We also have a press sequence. So this is achieved using the sim_press function. And the usage of this function is really similar to the 1pulse, except that we now have echo time parameters that we have to supply as well. If we look into the actual code of this press sequence, it's also similar to the 1pulse experiment, except that we have an excitation pulse followed by 2 180-degree rotations with little delays in between as well. And so we can also test this out on the lactate spin system. So in this case, I'm generating a series of echo times from 20 milliseconds all the way up to 300 milliseconds. And I'm going to simulate the press sequence using those different echo times using a for loop. And I'm putting my outputs as a cell array. So each different echo time appears as a different element of the cell. And because the op plot spec function accepts a cell array of structures, then we can plot them all at the same time using OP plot spec. And what you can see is the lactate spectrum using the press sequence for echo times ranging all the way from 20 to 300 milliseconds. Now, the simulations that I've shown you so far are all done using ideal RF pulses, meaning that the RF pulses are assumed to be instantaneous. But we know that these type of simulations don't agree well with in vivo experiments, because in vivo the RF pulses are not instantaneous, they have a shape to them. And so we need to take this shape into account and take into account the chemical shift displacement that's associated with the RF pulses in order to do accurate simulations. And that's what I'm going to show you next. So when we have a shaped RF pulse, we know that this shape is going to lead to a particular frequency profile, and that frequency profile, when combined with a gradient, is going to lead to the selection of a slice. Okay, and it's this gradient here that creates a mapping between position and frequency and allows a frequency selective pulse to then generate a slice selection. The problem is this mapping actually depends on the chemical shift of the spins that you're talking about. And in spectroscopy, we're looking at many spins with different chemical shifts. And what that means is, whereas one spin might result in a slice selection at one position, if you have spins with different chemical shift values, their selected slices are going to end up in different positions. And so this is something that our simulations really need to take into account in order to do an accurate job. And in the case of a press sequence, the situation is a little bit complicated. So we have two refocusing pulses that are applied in different directions, and it's the intersection of these two refocusing pulses slices that is going to give us our region of interest. Okay, so this is what the region of interest might look like for one spin. We're calling it spin A. It's the intersection of the two slices generated by the two press refocusing pulses. But for another spin system with a different chemical shift, those two slices are going to be shifted. So the same refocusing pulses are going to generate slightly shifted slices for our other spin, which we're calling spin X, and the intersection of those two is going to be in a different position. Now, if I'm simulating the signal from spin A, I need to look at the signals that are generated from inside this first box here. So what I can do, I can eliminate all of the other regions, and what I need to now do is break this system up into it looks like 4 different regions, and I can number these regions 1 through 4. And in region 1, this is the region where the coupling partner of spin A, spin X, is actually experiencing neither of the two refocusing pulses. In region 2, the coupling partner spin X experiences only the second refocusing pulse. In region number 3, the coupling partner spin X experiences only the first refocusing pulse, and in region 4, the coupling partner is experiencing both refocusing pulses. And it turns out that the signal that we observe for spin A depends strongly on whether or not its coupling partner experienced these refocusing pulses, and so we really need to take this into account in our simulations. The consequences of this are that the spectral shape varies within the region of interest, and this is true for both standard press and megapress experiments, and accurate simulations must take this information into account in order to accurately estimate the spectral shapes. So how do we do this? We account for this by repeating our simulation at an array of spatial positions. So if this blue box indicates our region of interest for the spectroscopy exam, we're going to simulate what the spectrum would look like at each one of these positions. And at each position, we're going to take into account the exact shape of the RF pulse and the gradients that are used for slice selection. And then at the end of this, we're also going to try and remove some of these outer volume signals using crusher gradients or by phase cycling. And then our total signal is going to just be the sum of all of these individual simulations added together. For an example of a FID-A simulation function that uses shaped RF waveforms, we can look at the run sim press shaped function. This is a press sequence with shaped refocusing pulses. And maybe the best way to look at it first is to actually look at the code. And what you see is a list of parameters that you need to specify before running the simulation. And so the very first parameter is the refocusing pulse waveform, but also you need to specify things like the field strength, the size of the voxel, the grid size. So here we're doing a 16 by 16 grid. And also you need to specify the echo time. Here we've chosen 135 millisecond total echo time, and the spin system here, which is lactate. Once you've chosen all those parameters, you can save the script and run it using just the name by typing the name of the script at the command line, which is run sim press shaped. Then the script will run, and it usually takes about 3 minutes to run, at least for these parameters that we've chosen. And it's going to produce two important outputs. One is the spatially resolved outputs called out_pos_xy, which has the simulation at each spectral or at each spatial grid point, and the other result is the total sum of all the spatial positions, and that's just called out. And I can plot both of these. So for the spatially resolved output, I can use a function called make2D simplot, and in this function I just pass in the spatially resolved output and the frequency range that I want to plot on. So in this case, I want to look at the 1.3 ppm lactate resonance. So I'm just going to plot from 1.1 to 1.5 ppm. And when I run that command, I'm going to see a spatially resolved plot of the lactate as a function of position. And you can see the selected region of interest is sort of around this box area. And you can also see that the 1.3 ppm lactate peak is mostly inverted throughout that box, throughout that region of interest. But in some parts of the region of interest, it's actually not inverted at all, it's upright. So that's the kind of modulation that I was telling you about earlier. If we want to look at the 4.1 ppm lactate resonance, we can use the same function, but just change the frequency range that we're looking in. And when we do that, we can see clearly that again, the lactate signal varies throughout the region of interest. And we also have some chemical shift displacement so that the 4.1 ppm signal doesn't come from the same location as where the 1.3 ppm lactate signal came from. We can also view the total result after summing over all of these spatial positions if we just use op-plot_spec out, which is the summed output. And when we do that, we see the total, which is the accurate estimate of what the lactate signal would look like under an experiment like this. And you can see it's inverted at 140 millisecond echo time. To give you an idea of how accurate these simulations can be, we did some experiments with phantoms, and we compared those phantom experiments to simulations. And we did that at short echo time using a press sequence, and also at longer echo times using a press sequence. And you can see very good correspondence between the simulated spectra for GABA and the experimental spectra. We also did this using glutamate, and again, you can see very good correspondence between the simulated and experimental glutamate traces at both long and short echo times. Lastly, what I showed you before was simulating the simple press sequence, but since we're at the editing school, I thought it would be good to show simulations for an editing experiment like megapress. So in order to do this, we need to simulate both an edit on and an edit off experiment, and then we have to subtract them. FID-A has a few built-in simulation scripts for MegaPress, and you can use both non-spatially resolved simulations using these two functions, but you can also do spatially resolved simulations depending on the level of accuracy that you need. And in this example today, I'm just going to show you the function run sim megapress shaped edit, which is a function that does not include spatially resolved simulations, but it does take into account the exact shapes of the editing pulse. And if we once again look at the code of this run script, we can see that there's a list of parameters that we need to edit before we run it. So we need to specify the shape of the editing pulse waveform, and we need to specify the frequency in ppm of both the edit on pulse and the edit off pulse. We also need to specify the field strength, some of the timing parameters of the pulse sequence, and the spin system that we want to measure. And this is the run sim megapress shaped edit function. This function is calling another function called sim megapress shaped edit, which shows you the actual sequence of operations on this pulse sequence. So we have an excitation pulse, followed by the first refocusing 180 pulse, followed by a shaped editing pulse, the second press 180 pulse, and another shaped editing pulse, followed by the readout. And if I run this simulation, so I'm going to call the function run sim megapress shaped edit from the command line. And when I run that command, it takes a little less than a minute to run. And the main outputs of interest are the edit off spectrum, which are called out off, and the edit on spectrum, which is called out on. I can plot them both using op plot spec and passing them together as a cell array. And what I see when I do that is the edit off spectrum, which is this blue one, and the edit on spectrum, which is the red one. And if I want to look at the difference spectrum, I can subtract one from the other, and I do that using the FID-A function op underscore subtract scans, and I subtract edit off from edit on. And when I do that, I get a new spectrum called diff, and if I plot that using op plot spec, I get the simulated difference spectrum, and this is the sort of familiar shape of the J-difference edited GABA signal from a megapress experiment. Now, the last thing you might want to do after running these simulations is you might want to export your data into LC model format so that you can make a basis set. So first of all, when you're making an LC model basis set, it's a good idea to use the most accurate possible simulation given your time constraints. So typically, I recommend using a spatially resolved simulation with fairly high resolution. That is a grid size of at least 32 by 32, and that you should be using the exact pulse waveforms and timings that were used experimentally in your in vivo data. Once you've generated a simulated spectrum that you're happy with, you can write it into the lcmodel.raw format using the io_write LCM raw function, and that will take your final simulated spectrum, here we're calling it diff, and turn it into a text file called gaba.raw that you can then use to import into a basis set in LC model. This is just for one of the metabolites, but what you should do is repeat this simulation for every metabolite that you want in your basis set, and then export each one as a .raw file with its own metabolite name. And once you've collected all the .raw files on your LC model server, then you're going to use the LC model command make basis to convert these .raw files into an LC model basis file that you can then use for LC model analysis. So that's the end of my talk. I want to thank you for your attention, and Just to summarize, the FID-A toolkit provides a command line environment for MRS data processing, simulation, and basic RF pulse design. It gives the user a high degree of control and flexibility and efficiency. And because it's open source, it provides users with a possibility to build upon the existing tools or to change them to suit their needs. So thanks again for your attention. Thank you to everyone in my lab and to the collaborators that have helped me over the years, and also thanks to my funding sources. So I guess I won't be able to take questions right now, but I think there's going to be a live discussion forum later in this meeting, and I hope to see you there, and I'll be happy to answer any questions you might have at that time. Thanks very much.